Thank you so much. Yes, today I'm delighted to share how our team at the University of Colorado, in collaboration with colleagues at the University of Chicago, have adapted nanopore sequencing in order to measure and read out intact aminoacylated tRNAs, addressing a key gap in measuring the supply of the functional raw materials for protein synthesis at the molecular level. And so this approach enables direct measurement of aminoacylation while preserving sequence and modification information, a combination not captured by prior tRNA sequencing methods. So just as a bit of background, tRNAs are the fundamental adapters of translation. They link codons on mRNAs to amino acids in protein synthesis. But while Nobel Prize-winning structures of ribosomes like this one have provided groundbreaking insights into how mRNAs are translated into proteins, until recently, when it came to directly reading out the functional supply of charged tRNAs, which are the actual substrates required for protein synthesis, we faced major limitations. And so one of these is that tRNAs are highly modified, and this means that cDNA-based tRNA sequencing methods that rely on reverse transcription often stall when they encounter these modifications and struggle with bias. In addition, while nanopore direct RNA sequencing has given us a lot of new insights in the past couple of years about tRNAs, a major gap was our ability to directly resolve aminoacylation. And this is the chemical linkage that defines tRNA charging. And so at the outset of this project, there was no high-throughput method to directly profile charged tRNAs at a systems level, leaving a critical gap in our understanding of translational control. So to prepare tRNAs, aminoacylated tRNAs for sequencing in this method that we call aa-tRNA-seq, we developed a two-step ligation strategy. In the first step, charged tRNAs are selectively captured at their 3' ends via chemical ligation to the amino acid, guided by a short 5' splint adapter. And then in the second step, we enzymatically ligate RNA adapters onto all of the free ends of tRNAs in solution, be they charged or uncharged, that are remaining after this chemical ligation. And so all of these molecules are then directly competent for nanopore sequencing using the direct RNA chemistry. And this allows us to recover both charged and uncharged tRNAs in the same pool. And this preserves not only the aminoacylation status, but also sequence information and modification information. And the resulting library is competent for nanopore direct RNA sequencing, and this gives us single molecule resolution of tRNAs and their charging across the entire tRNA pool in a cell. So I said this approach relies on a chemical ligation reaction, and this forms a stable covalent bond, a phosphoramidate linkage, directly between the 3' amino acid and an RNA adapter, and this preserves all the way through to sequencing. The chemistry is efficient, it's also selective. So on the right-hand side of this slide, I'm showing you a very laborious method called an acid northern blot, in which you can see that our adapter on the right only reacts with charged tRNAs, creating this shift. After adapter ligation, aminoacylated tRNAs can be sequenced directly using standard Oxford Nanopore direct RNA library preparation and platforms. So here I'm showing an example comparing synthetic tRNAs that we charged with one amino acid, glycine in orange, compared to an uncharged tRNA control. We aligned the sequence signal across the 3' end of the tRNA, including the CCA tail, as well as across the adapter sequence. And so here I'm just showing you this signal difference in terms of the mean current readout for these molecules across the 3' end of the molecule. And what I hope you can appreciate here is that first, there are some real signal differences in the ionic current as we pull through aminoacylated tRNAs versus uncharged controls. And second, that this signal actually covers a fairly broad footprint. So it's not just a single position. The dashed line indicates the actual location that is aminoacylated, but we have these broader footprints that provide rich information that we can use for downstream classification. However, the magnitude of this shift varies. This varies both depending on what the amino acid is, and it also has some variability across different tRNAs in our sample. So this means that traditional base calling or cutting a single value threshold really isn't sufficient in order to distinguish charged from uncharged tRNAs. So instead, we trained a machine learning model directly on the raw signal in order to robustly classify aminoacylation state. And so to classify charged and uncharged tRNAs directly from nanopore signal, we turned to budding yeast and we prepared 2 libraries for training. First, we did one using only chemical ligation to aminoacylated tRNAs. And then we prepared a second library from chemically deacylated tRNAs. We stripped the amino acids off and we did only enzymatic ligation to these. And these gave us 2 ground truth libraries. And then we went and we trained a remora model looking at a 6 nucleotide training window around the aminoacylated site. And when we did this, we were able to get a model that distinguished quite nicely between charged and uncharged tRNAs with high performance, achieving an F1 score of 0.966 and 0.98 on held out test data. And this gives us a robust and scalable method to detect tRNA aminoacylation status at the individual read level. We then applied our model to a native mixture of charged and uncharged tRNA extracted from budding yeast grown in rich media. And here I'm comparing the percent of molecules classified as charged by sequencing on the x-axis to independent measurements obtained from acid northern blots on the y-axis. And while sequencing underestimates absolute charging, primarily because charged molecules are translocated more slowly through nanopores, the relative trends are strongly correlated, and this has enabled us to systematically profile aminoacylation across tRNA families in biological samples. So we then validated our method under a nutrient stress known to produce biological changes in tRNA charging. So we subjected a leucine auxotroph, budding yeast, to 15 minutes of leucine starvation. On the left, this is a simple chemical charging northern blot that allowed us to look at one specific leucine tRNA where we observe an approximately 28% drop in tRNA charging. When we put this same sample through our sequencing workflow, shown at right, we can systematically measure tRNA charging across all tRNA isodecoders, not just one in this low throughput format. And what we see is that the Lucille tRNA isodecoders in this leucine starvation experiment show some of the biggest statistically significant drops in charging. Most other tRNAs remain unaffected, although surprisingly we did observe several alanine tRNAs that hadn't previously been reported and that actually go up in tRNA charging under these conditions. And this underscores the power of our method to reveal subtle and dynamic changes in translational supply. And furthermore, our synthetic tRNAs that we charged with individual amino acids allow us to profile the signals produced by each individual amino acid during aa-tRNA-seq. So we observed that these amino acids produce reproducible and systematic shifts in both the dwell time and the ionic current as we pull these molecules through nanopores. On the left, I'm showing you the mean dwell time for synthetic tRNAs charged with each of the 20 standard amino acids ordered by side chain mass. On the right are changes in mean ionic current relative to an uncharged tRNA control. And one thing that stands out to us as we look at these is that we see the largest changes in dwell time as the amino acid is actually interacting with the motor protein, as it's forming contacts with the helicase transiently. And we see the biggest changes in current when the amino acid itself is within the reader head of the nanopore. So these results demonstrate that amino acid identity imparts a physical, detectable signal, and they lay the groundwork for classification approaches based on nanopore signal properties. So this is a valuable ground truth data set for model training. And so since we had established that amino acid identity produces distinct nanopore signals depending on the amino acid, we next trained models to classify these using our synthetic tRNAs charged with individual amino acids. So we again trained another remora model over a slightly different window with pairwise comparisons. And across these, most models achieved F1 scores greater than 0.85, confirming the robustness of these signals. So we're excited about this method. We believe that direct measurement of charged tRNA pools opens up new opportunities in a number of different areas. We think that in research, this method will enable researchers to systematically profile supply and demand during translation under different conditions. In synthetic biology, these kinds of insights may give us new ways to do codon optimization and to engineer tRNA pools for optimized expression. And then lastly, in the therapeutic space, we think that understanding tRNA supply, and particularly the supply of functional charged tRNAs available for translation, could inform new strategies for targeting translation and disease. So we've built this entirely on ONT's commercially available platforms using standard RNA flow cells, direct RNA chemistry, signal analysis tools like Dorado and Remora. And this allows us to be downstream compatible with changes in base calling and ongoing improvements. So with that, I'll really just close and say we're excited that this method expands the molecular resolution of direct RNA sequencing, moving beyond nucleic acid sequence alone to directly profile the functional translational landscape. Combined with rapid developments in nanopore-RNA modification detection, this brings us closer to a full systems-level view of translational control. I should also thank the team behind this work, especially my co-first author, Alex Radakovich, who kicked off this project as a PhD student in Jack Szostak's lab, as well as all of our team at the University of Colorado in Jay Hesselberth's lab. So our full method and results are available on bioRxiv, will be published soon, and we're excited to continue building on this foundation with the community. Thank you all so much.